guys, this is Alicia with Good Morning Sunshine. So today was vet school day 3. Today I had 4 hours of lecture, and then I had my equine lab today where we learned how to handle a horse and put a halter on a horse and know what side to come from and be very safe about it. Big animals is not my thing, but the lab went okay, so that was nice. And 4 hours of lecture though, so a lot of material, a lot of different material. I think we went from embryology to like gene expression. I don't know. Oh, the cell cycle. I don't know. Just a lot of things. So I was my friend actually texted me saying she would love pictures of me when I'm explaining things. However, I will be doing that soon enough. I'm actually waiting for my desktop and desk to be put up, so it'd be much easier so I can teach you that way, because I'll have a really big screen. It's hard when I just have my laptop because I need to look off notes and stuff too. But yeah, so like things I tell you I haven't really studied yet. It's just from what I've learned in class and what I've retained from class, but then I want to see how much I know on video, which is probably not a lot, so, but then it tells me what I don't know, kind of thing. But yeah, I'll start with embryology. We were just learning the basics of it, a basic overview of it. So there was that. So we have to basically assist in understanding certain gross anatomical features of embryology, and that's what she was trying to teach us. I think I get really confused because they're trying to teach us all this stuff, but just like the generals of all this stuff. So I'm like, I really don't know what we're supposed to know because it's very vague. But yeah. So in the end, though, we have to provide background for understanding the development of certain common things about embryology, I guess. So anyways, let's move on. So she started with a few definitions, one being primordium. Sorry, that's a hard word to say, but it's a primitive structure that will eventually give rise to other structures. So like think of an embryo, I guess. This tiny little egg starts as an egg with many cells. That's all it is. And that's the structure, basically the structure of a Yep, I'm going to stop saying it because I can't say it. But anyways, and then the next definition was presumptive, which is a primitive structure that will eventually give rise to other structures of a more complete and recognizable form. So again, it's not just the embryo anymore. It's going into structures that you can now recognize. And then we have the definitive, which is a term used to describe a structure once it has attained final form. So think of it like a baby fetus. Now you're in your final form. Born. So there was that. I kind of got that. And then we went to the basic mechanisms of development. So let's see. What did we learn on that? So basically we're trying to get from a single cell, tiny embryo, to a topotent cell. I'm probably saying that wrong, but that's of a relatively simple unspecialized structure, and then we're trying to get from there to the total genome, which is comprised of billions or trillions. So like in the human body, we're composed of over 10 trillion cells. Whales have even more cells. Horses have even more cells than we do. So, but we have to remember that each one of those cells has a complete and total genome in it. Even if it doesn't express everything, it still has a total genome. But it only expresses a tiny portion of it. So I think that was the main point she wanted to get across, that every cell does contain the whole genome. Let me see. And then we got into basic terms, I guess. Growth is an increase in size, obviously, and it has 3 basic mechanisms, which is the number of cells, the size of cells, and the amount of extracellular matrix. And she's like, I know this is obvious, growth is obvious, but I still have to teach you it. She's really funny. I actually like that professor. I think her name's Dr. B. I think. I could be wrong. There's so many professors, I don't know. So then next we got into patterning, which is the establishment of correct orientation or position of pre-programmed subgroups of cells to each other or surrounding tissues. So it usually precedes differentiation or morphogenesis. But patterning is very important. If there's an error in patterning, then it could all be bad if there's a mistake in patterning. We can get things such as a hairy eyeball. And what do we do about that in an animal? Well, she said we just take the hair off the eyeball and it should be okay, as long as it didn't disrupt the cornea or anything. But yeah, it could lead to some very weird stuff, she said. She sort of showed us some really cool pictures. I can show you the hairy eyeball one. Can you guys see that? But anyways, that's the hairy eyeball, and then it getting removed. So that happens when there's a disruption in the patterning of cells. And then we got into differentiation, which is the process by which similar cells in a population become different from each other. Or each cell in a population expresses a different phenotype. And then this process is gradual, stepwise, and controlled. And then we got into morphogenesis. I know my laptop doesn't want to work right now, so that's fun. But morphogenesis is any change in just the shape of cells or tissues. I'm just going to hold my phone because it's easier. Or is it a massive cell that changes to produce a structure with a defined shape? So it's a little different than differentiation. And I love how we went from morphogenesis to cell death, basically. So that's apoptosis, which is programmed cell death. So some cells can actually be programmed to basically die. So like some horses, I guess, have extra toes, but then they have those cells go through apoptosis. I always say it wrong, doesn't matter. But anyways, and then that's how their toes get removed. Also, another example would be we have webbing in our fingers, and if apoptosis doesn't take place, and then we would have webbed fingers. But it does take place, so at a certain point in time, we get rid of our webbed fingers. So I thought that was really cool. Oh, here's that example. Here's that picture. So we have webbed fingers here, and it goes through apoptosis, so that's why we do not have webbed fingers. And then here's the horse's example I was talking about. So they lose 4 or 5 original digits due to apoptosis. And then she kind of skipped some stuff. And then we got into induction, which is a sequencing mechanism. It ensures an orderly sequence of events, culminating in development of a highly complex structure. So you have an inductor and an inductee. So one population of cells, which would be the inductor, acts on another inductee to produce a change in behavior of the second group in some developmentally significant way. So let's see. So basically, induction is a mechanism that ensures an orderly sequence of events culminating in development of a highly complex structure. I know some of this is hard to make sense because we just get a brief overview as well, so I'm sure it sounds like jibber jabber to you guys. But yeah. But it really helps me because I need to learn this stuff too. So yeah. She writes in bold here that the action and or presence of one tissue is necessary for the normal development of another. So I guess basically if induction doesn't happen, nothing's going to happen, and that's a problem. Inducing capacity is usually localized. Timing is very important, and strict sequencing is essential. And inductees must be differentiated enough. That's what that meant. Alternating cycles of induction and morphogenesis mechanisms are not well known. And I believe the inductee has to be by the inductor in order for this to take place. So oh, and then she got into the notochord. So the notochord is the red thing right here, and it has to be close to the inductor in order for anything to happen. So then induction would then take place, and then after induction takes place, like on the ectoderm here, morphogenesis comes into play, which I gave you the definition of that earlier, and then it can go through many morphogenesis is that a word? I don't know. But anyways, yeah, there was that. And then we got into interdependence and interrelation of processes. That's a lot of big words to me, but basically it means that all processes depend on each other. So differentiation depends on morphogenesis, which gets into inductors, inductees, and the notoderm. So all of these things rely on each other for everything to work out properly. If something goes wrong, it can go very wrong. And then at the very end, she threw in the definition of mesenchyme, which is the relatively unorganized connective tissue of the early embryo that fills much of the space within the embryonic body. So this is what mesenchyme looks like here. But it was just kind of thrown in, so like I don't really know what it does, but it gives rise to connective tissues of the adult, like blood, bone, and cartilage. It controls the ectoderm and eventually the epithelial overlying it. So like, I think she was trying to explain it, but with a chicken foot, like if you have a chicken foot and you put it with a different kind of, like a frog, it will still make a chicken foot on that chicken, even if it had frog epithelium. I don't know. It was something weird that I'm trying to remember. And then we had conservation of the genome, which is the uniqueness of each species is related to the uniqueness of its genome. But the genome is often conserved to a far greater degree than one might seem. Yeah. So that was the end of that one. And then, let's see, what else did we get into? That was just the first lecture, the first hour. And then we got into regional differentiation of germ layers. So I guess that kind of made sense, going from the embryology. So then we got into cleavage. What is cleavage basically? It's early cell divisions take place with an increase in cell number, but a decrease in individual cell size. So I also took notes on this, and I wrote some things down. But why does this happen? Why is there an increase in cell number, but a decrease in the individual cell size? Well, I wrote here that a zygote has a total genome. So remember how you talked about cells always have a total genome inside them. But one nucleus can't control everything in that much of the cytoplasm. So the nucleus needs less cytoplasm to control, thus decrease in cell size. And first, a solid ball of cells forms, which later becomes hollow. And the hollow ball of cells, what I just talked about, is a blastocyst. blastocyst. There we go. And then, sorry guys, this takes forever. So the blastocyst consists of an outer cell mass, which is the trophoblast, and then it also has an inner cell mass, which is the embryoblast, which makes sense. Embryo, inner, troph means outer. Yeah. But basically, the blastocyst is the developmental stage in which the conceptus is a hollow, fluid-filled sphere, kind of like a balloon with a small bump stuck to one inside edge. I guess I can show you this picture. So at different cell stages, but here's an early blastocyst, and this is a late blastocyst. I don't even know if you guys can see. I hope you can. Because I still don't know how to switch the camera. But here's an actual blastocyst. So it's hollow. So the outer cell mass is basically a thin layer of the cell that surrounds the embryoblast, because the embryoblast is the inner part. Let's see. So then we got into gastrulation. Pretty sure that's how you say it. Which we have the ectoderm, which is the outer layer. I'm sure you've heard these terms. The mesoderm, which is the middle layer, and the endoderm, which is the inner layer. I said mesoderm is the middle layer. They just sounded the same for some reason to me. So each of these three germ layers produces primordia of specific types of definitive tissues. So then we got into talking about embryoblasts specifically. So the formation of ecto and endoderm, embryoblast itself differentiates into a superficial and a deep layer. So the ectoderm being the superficial layer and the endoderm being the deep layer. And then we got into the formation of the mesoderm, which we talked about the primitive streak, which is the center of a cell, proliferation and movement. It's the primitive streak's function is to form the mesoderm, which the mesoderm member means middle. It is a streak formed by cellular proliferation and migration, producing two elevations with a depression or streak between them. Here, I'll show you a picture actually. So basically, here's the picture. I hope you guys can see because I really don't know what I'm showing you right now. But anyways, this is the streak right here in the center right here. The primitive streak this is the dorsal view of the early embryo. So this is an embryo and then there's the streak. It looks funny, huh? I thought it did. And then when the mesoderm has fully formed, the primitive streak should completely regress, like it should go away basically. There should be no primary primitive streak tissue left. So then we really got into the nodal cord, which is the inductor for the head and CNS, and it's an organizer for the vertebral bodies and some basal skull bones. But yeah, it forms at the end of the primitive streak, so that's why we went into the nodal cord. So I wish I could show you pictures, but so here is the primitive streak right here. I don't know if you can see it, but it's orange right here, and then it turns into, and then the nodal cord processes is in front of it, which is fun time. The nodal cord is basically a column of cells that grows cranially from the cranial end of the primitive streak, like I said. Yes. So this also normally regresses almost entirely, leaving only the nucleus pulposus of the intervertebral disc. So it's like the mushy part of your vertebral discs. I'm sure you've seen bones of that. So basically that's the groundwork for organogenesis. These are all big words to me. So with the establishment of all three germ layers, now the necessary tissue types are present and begin formation of all body systems. So all of that had to take place in order to get to the point where they're making body systems. So again, we have differentiation of the germ layers, the ectoderm, mesoderm, and endoderm. So the surface ectoderm is part of the skin. It's specific parts of the ear and eye. The neural ectoderm is for neural tissue. And then we have neural crest cells. Which are a highly specialized group of cells that eventually give rise to disparate tissues with extremely specialized functions. Yeah. Let's see. My computer's being very slow. I talked about that. Sorry guys, you have to hang in there with me for a minute. She got into the discussion of between optic and otic, between ears and eyes, and if we misspell something on test, she's really gonna ding us for it. So there's that. So then we got into early differentiation of the mesoderm. It's initially a thin layer, fairly evenly distributed between the endoderm and ectoderm. It undergoes characteristic specializations in particular areas. It undergoes morphogenesis in three regions, which is the periaxial, the intermediate, and lateral. The periaxial mesoderm is paired columns of cells, lateral to each side of the nodal cord. I was gonna see if I can bring up a picture here too, but sometimes they don't give us pictures either, so that makes it even more confusing, I think. Yeah. Let me see. So the mesoderm is number 2 here, is that orange spot. And then it eventually, like the 3 regions I was talking about. So we have the mesoderm here, we have the intermediate here, and then we have the lateral mesoderm on the sides and the periaxial. I hope you guys can see it because I'm trying my best here. But all this stuff was really confusing. So the 3-dimensional columns basically eventually break up what I was just talking about, the periaxial and all that. These end up breaking up into chunks of tissue, so which becomes somites. So this becomes the spine. There, if that makes any sense. It kind of makes sense to me, but not really. Let's see. And then somites, each somite forms distinct regions. So we have a lot of different regions. We have the sclerotome, which are cells from ventral and medial walls of the somite. They surround the spinal cord and nodal cord to form vertebrae and ribs. And then we have the dermomyotome, which is cells of somite's dorsal wall subdivided into 2 parts. So this has 2 parts. There's the dermatome, which is the more external cells spread under the ectoderm to form the dermis of the skin. So the dermomyotome is basically the formation of parts of the skin. And then we have the myotome, which is a more internal form, epaxial skeletal muscle. And then we had the syndiotome, which are for tendons and ligaments. I wish I could show you these. Sclerotome. We didn't really get pictures of these either. So that kind of sucks because I wanted to show you guys. Or they're like pictures you wouldn't understand, really. Because I barely understand them too. And then we got into the intermediate mesoderm, which are kidney precursors. They create the kidneys, the gonads, and the tubular structures of the reproductive tract. And then we have the lateral mesoderm, which is the splitting of lateral mesoderm. Oh, it has 2 layers. I don't even know why I was talking about that. It has the somatic mesoderm layer, which is the superficial layer, and then we have this splenectic, which is the deeper layer of, basically, yeah. See. Oh, and then we got into the early differentiation of the endoderm and the formation of the GI tube. It has 2 body foldings. It has the head fold and the tail fold. The head fold is the elevation of head process, formation of the foregut as the folding progressively confines the cranial part of the yolk. The yolk sac. Let's see. So basically this goes over folding. Here's the yolk sac, I guess. And there's the connection. I don't know what any of that means. So that's going to be something I have to study because I have no idea what that means. And then we have transverse folding, the lateral body folds, the growth of somites, pushes body wall laterally and ventrally. So that's what I just showed you in that picture, whatever that means. We'll get there. We will get there. So the ectoderm is mainly the epidermis of the skin. So then we got into, I don't even think she got this far, to be honest. Yeah. So that was it for the second hour of lecture, which again was a lot. And then we got into intracellular communication of ligands and receptors. So this professor's kind of hard to understand for me. I don't know if it's his teaching style that I can't wrap my head around. I don't know. I'm trying though. So that's all that matters, right? So he got into sensing and signaling systems. We got into nomenclature. He just wanted to give us examples of ligands, what they do, how they are signals basically, and then how that ligand goes to a receptor, which then the receptor goes to secondary messengers, which then enforce a signal transduction. So basically he just gave us all different examples, and he wants us to know that things happen. Like when a receptor receives something, it goes to a messenger, and then something happens, a signal happens, and something takes place in order to fix things. So he just wanted us to wrap our head around that. Basically, that was the whole lecture. Like, we have to know about activated ligand channels. And then, what are classic second messengers? Like, we have CAMP and ATP and calcium. We have to know simple diffusion, protein, phosphorylation. I'm not going to get into this with you guys because we have to get into G-protein coupling receptors, basically how an EpiPen works. But yeah, he was just giving major examples, he said we only need to know the general idea of them, which I just told you. But he just wants to know that things send something, send a signal, and things happen, basically. So I'm not going to get into too much detail because we're already at 30 minutes. And yeah. So then we got into gene expression, the cell cycle, and programmed cell death, which I kind of already talked about. We got into the central dogma, which everyone should know. DNA is for replication, DNA to RNA is transcription, which is nucleotide-based pairing, and then RNA to protein, which is translation, is the 3 nucleotide units coding for amino acids. And then, so the central dogma we've already learned a lot about. And then we got into RNA transcription. It involves the action of an enzyme complex, an RNA polymerase that can assemble an RNA polymer whose sequence corresponds to the coding strand of DNA. And then we got into gene expression regulation, which is regulated by families of transcription factors, proteins that bind to specific DNA promoter regions and either prevent or allow transcription to occur. So then he gave examples of that too. And then we got into the cell cycle, the steps of the cell cycle. He said we didn't really need to know the steps of the cell cycle, just that things can happen where the cell cycle has to be stopped. So you remember interphase, prophase, prometaphase, metaphase, anaphase, and telophase. So basically, key points were regulation of gene expression and mitosis, including entry into or progression through the cell cycle occurred by mechanisms similar to those in other signaling pathways, and that the process is closely monitored for errors, because if error happens, that's unfortunate. We got into transcriptional errors, change in the amino acid sequence, the mutations and other regulatory components, the magnitude of that change, and the function of gene whose expression has gone awry. So things can happen at all steps of the cell cycle, basically. But it has a proofreading function. So there's consequences of DNA damage, but DNA damage can be detected by specialized pathways in the nucleus. Transduction systems exist that resemble those signaling from outside the cell. The consequences vary from DNA repair to activated cell death, which is apoptosis. So there is always that option in case things cannot be fixed. But there, like I said, the cell cycle has many checkpoints. It has M-phase checkpoints as well, S-phase checkpoints, which are spindle checkpoints. And then we got into apoptosis, which I talked about earlier. Yeah, I think that was it for today. I have my bovine, which are cattle lab tomorrow, which I also have an anatomy lab tomorrow, and there's one more lab too that I have. Microscope lab. So all I have is all day tomorrow is labs. The last one being the cattle lab, which apparently people can pass out because we're in 90-degree heat in our clothes, covered by our coverall, in rubber boots, in 90-degree weather for 2 hours. So I'm kind of worried about that. We're said to drink lots of water, bring lots of water. So that's the main lab I'm scared about. The equine lab went great today, so at least I got that one out of the way. But yeah, tomorrow's just full lab. At least there's no more material because I am drowning in material right now. As you can see, it's already 34 minutes. Please you don't have to watch this whole video. It's just for my reference. So I have notes set in my voice too, as well as having the PowerPoints, as well as having my condensed notes. So it's just different ways for me to look back and learn. But I thank you guys for watching. I try to be better about pictures. I swear when I get my desktop, it'll be better so I can really show you what's going on. Even if I don't know myself, I think pictures would really help. So I love you guys. I hope you guys had a good day today. It was a long day for me, but hopefully not a long day for you.